So I had the perfect setup from George, because what I basically wanted to start out by saying is there are these established nanoindentation techniques that are very common for elastic plastic materials, for hard materials, for metals, for ceramics. Occasionally you can kind of use them on other types of materials, but and this slide actually looks exactly like George's. I think we're using the same schematic of the indenter even, the same equations, the same basic graph. This is for elastic plastic materials. And if you want to look at biological materials, if you want to look at hydrogels, if you want to look at a lot of interesting materials that are not elastic plastic, then you have a problem if you just take these approaches for elastic plastic materials and apply them directly. So in fact, the very first thing that I did in the field of indentation testing was looking at time-dependent deformation. So if you have something like cartilage there on the left and you go to a constant indentation depth, you get dramatic load relaxation because the material is 80% water and that water starts to flow. Similarly, if you look on the right, there's some data for creep in bone. So that left scale is displacement, and you're looking there at, you see loading up in some 20 seconds or so and then sitting at a constant indentation force, and you get creep. And again, this is due to fluid flow in the bone, can also be due to viscoelasticity in the solid matrix. So we did a whole series of studies. In fact, Ginger was one of the co-authors on these series of studies that we did, where we looked at the effect of tissue hydration on the stiffness by making nanoindentation creep tests with spherical indenters. And we used a series of polar solvents to essentially dehydrate the tissue chemically, so as we still had it in a fluid, but we were changing the hydrogen bonding and the effect of the water. And so what you can see there is that as you go to increased tissue hydration, you see decreased stiffness by about an order of magnitude. And this is in bone, which is only about maybe 10 to 15% water. So this is a pretty dramatic effect for a material that is not nearly as time-dependent as something like cartilage that's 80% water. And when we started looking at the numbers that we got out of this, we were seeing that the effective modulus was somewhere around 2 gigapascals for these fully hydrated materials tested in water or in physiological saline, when we did wet testing of our materials using a spherical probe and a viscoelastic model to analyze the data. And that was rather startling, since the data on the right show what you would get if you were testing dehydrated bone using a Berkovich indenter, using elastic plastic data analysis of the sort that George was just talking about. And there we were seeing average values. You see I have 18 gigapascals for that particular data set. You can see around 20, 25 sometimes. So we've got an order of magnitude problem here. So in order to try and establish what was going on here, we actually decided to do some systematic investigations. Now, I'm going to be talking about bone for these studies, but I think it's an interesting approach to think about when you are talking about any material that is not elastic and plastic, that looking at how what you're doing is influencing the measurements that you think you're getting by how you treat the samples. Now, I'm not going to spend a lot of time dwelling on PMMA versus epoxy. That's a bit of a niche question about treating bone embedding versus including. We did try both conditions. But more importantly is what we did on the right. So we have some mouse bones, and we're going to take the same samples of bone, and we're going to treat them in these different ways, test them in these different ways, analyze the data in these different ways, and see what we get. So the very first thing that we did here was we took samples that were dry and that were fully hydrated, and we tested them with a Berkovich indenter and with a spherical indenter. And I'll note, because George did comment on this, we use relatively large spherical indenters to keep the strain small to make sure that we have no plastic deformation, whatever that means in bone, and I'll come back to that. So we're using relatively large sapphire probes, so they're much better in terms of roundness than what people get when they try to have diamonds that are relatively small. So then we're going to take these data, and for each data point, so for each wet, dry, Berkovich sphere, we're going to analyze the data using one of 3 different models that are appropriate for those data. Now, the important thing to note about these different models is that they are all based on the exact same elastic contact mechanics. So the fundamental equation of Hertzian contact for a sphere or of the Snedden solution for the pointy tips, that underlies all of these models. So we are not changing the elastic contact mechanics. What we are changing is how we are interpreting the inelastic deformation and whether it's viscous or plastic or some sort of viscoelastic. So I don't have to really belabor this point anymore since George set me up so perfectly. If you are using the elastic plastic data analysis based on elastic plastic contact mechanics, you use the unloading data, you use a sharp tip, you calculate modulus and hardness. And I'm really going to focus on modulus here. Now, you can have other ways of analyzing the data. So about the early 2000s, one of the first things that I did in sharp indentation, I've been working in indentation for more than 20 years, but one of the first things I did in sharp indentation was actually to just develop a simple viscous elastic plastic model. So basically just adding a time-dependent component to elastic plastic contact mechanics and using the full data set, so actually fitting the full curve, not just using the unloading, since the unloading, as you can see in the lower right there, when you have time-dependent deformation, the unloading isn't elastic plastic, and in fact it can bow forward, showing that creep. And so we actually fit to the entire data, and that gives us three parameters. It gives us the modulus, again, based on the same Snedden elastic expression, and then we get some sort of a hardness and some sort of a viscosity. This is a pretty toy model. It's just meant to give these sort of three different time-dependent deformation modes. The third way of doing things is to do a viscoelastic data analysis. And now we go to using this specifically for the spherical case where we know there's no plastic deformation, where all we're expecting is time dependence due to the fluid flow and the elastic deformation based on Hertzian contact. Now, of course, we can also analyze the unloading data with a spherical indenter using the Oliver Farr approach. And so we have something to compare to for this case as well. Basically saying, if we do these spherical indentations, are we thinking that the data are viscoelastic or are they elastic plastic? So the approach for doing viscoelastic contact mechanics, you use the creep data to analyze it, so not the unloading data again. And this goes back to some pretty nice stuff. It was originally done by Lee and Radok in the 1960s, and we did it for solid polymers in the early 2000s, and then also did it for bone. So both this approach and this viscous elastic plastic model were developed for polymers and applied to bone to just see what would happen. So the first result is that if you take these different cases and you just plot up what you get as the modulus, and you have that modulus, plane strain modulus, Berkovich, sphere, different types of data analysis, wet versus dry, well, you can see that we have a big difference. So the big differences between our Berkovich data and our spherical data, especially for the viscoelastic case, you see this order of magnitude difference in analyzing the same data taken on the same bone samples. Now, thing number one that's interesting is that dry bone is always stiffer than wet bone, and it's only by a little bit, and we'll come back to look at that a little bit more. But we see this systematic marching down as we change the data analysis and as we change the contact conditions, and this is really interesting to us. So what we tried to do to understand this a little bit better was actually to go ahead and from the properties that we get, assuming that we have a sort of series model, we can make an approximation of how those properties correspond to how much of the deformation is elastic versus plastic versus viscous. So what we're plotting here for the same data for the modulus in the upper left are the deformation components. So purple for the viscous deformation, green for plastic deformation, and then red for the elastic deformation. So you can see that in some cases, the elastic deformation for the corresponding data are actually kind of reassuringly similar, and that's really interesting and really nice. And what's really different is how we interpret the non-elastic data as whether it's plastic or viscous. And you can see this even more clearly if I take, and instead of plotting these as the total deformations in nanometers, but instead plot them as the proportion of the total deformation for that indent. So here you can see that if you have a Berkovich indentation tip, whether the data are for dry or wet bone, the viscous deformation proponent seems extremely small. Whereas if you go to the spherical data, you see more viscous deformation, and then you see this huge difference in the difference between how much viscous deformation you think you have in a viscoelastic model and how much plastic deformation you would have. And don't forget, these are extremely large spheres, so there shouldn't be any plastic deformation. So what you're doing is you're interpreting viscous deformation as plastic deformation, and that's causing a big problem. And so that's where the really big difference comes between the spherical data and the interpretation of the spherical data in this case where we know there really isn't plastic deformation per se. Again, whatever plastic deformation actually means in this case. Now, we can compare these data to the literature just to make sure that we're not doing anything crazy. And this is why we started to do this very systematic study, because people were always doing their one experiment and showing their one data set. And so there were data available for Berkovich dry, Berkovich wet, spherical dry, and spherical wet indents. And reassuringly, our data from the systematic study on the same bones are always within the range of what's been observed previously. So differences between different studies have really been about the differences in how people have done the testing, how they've interpreted the data. And one final note about this that I think is really interesting is that if you plot just the ratio of the wet to dry modulus, it doesn't stay the same depending on how you analyze the data. So you can have anywhere from about 60% to 85% of the modulus of the dry bone in the case of the wet bone. So the differences between wet and dry are coming out as different depending on how you test the data and how you analyze the data. So to us, this is an interesting cautionary tale, and I think a systematic study of the sort that we'd really like to see more of. As I said, we did it in bone, but actually we've been doing similar things in lots of different types of material. So I want to bring in one final parameter into play now, and this is the size of the indent. Because when we're talking about materials that have some fluid in them and that have some fluid flow in their behavior, then you are also seeing effects due to the porosity and due to the scale of the porosity relative to the size of the contact. And so this is our funny little picture here of if we vary the size of the contact, how does that affect the size of the zone that we're indenting in the material, and what pore fraction we're sampling, and how different pore sizes might be sampled depending on the size of the contact. And this arose, again, just because of the interesting structures. Bone has these sort of 7 different layers of hierarchy depending on what length scale you're at. And so people have really liked nanoindentation for testing bone, because it seems like you can really get at these material properties instead of if you're testing whole pieces of bone tissue. Like in a bend test, you have this complicated geometry, you've got this complicated porosity distribution, and so being able to go in with nanoindentation and test the solid material and try and really look for material property differences is probably one of the reasons that there have been so very many nanoindentation studies done on bone, although mostly on dry bone with a Berkovich indenter, with Oliver Farr data analysis. So it may be sort of hard to interpret them. So we started to do some experiments to look at this length scale effect. And in fact, if you look at the picture on the right, that is a slab of gelatin. So in fact, we started doing these length scale experiments for the purpose of looking at hydrogels. And my second talk tomorrow will be about hydrogels. And so I'll reintroduce these experiments. But once we had developed this sort of parallel system for comparing nanoindentation data to data done using spherical indentation, but at a larger scale, we actually started looking at everything with this, because it turned out to be something that gave us a lot of information. So here we've got our setup for nanoindentation, and I have it in quotes because we're doing indentation depths that are micron scale, and we're using tips that are huge. So our spherical probes, as I said, are relatively large because we don't want plastic deformation. We want to have relatively small strains. Then we have things called microindentation, which is when we use our very small scale instron tester with a 5 Newton load cell, and we use tips that are on the order of millimeters in radius, stainless steel ball bearings that have been smoothed, and then we do indentation depths that are just sub-millimeter. So you could really say that we're doing microindentation and milli-indentation, not really nano-indentation and microindentation, but we use a nano-indenter machine, and so therefore we call it nano-indentation. Now for these data, I'm going to evaluate them in a poroelastic framework, and I'll have more to say about this tomorrow, and I know there are other speakers who will have more to say about this tomorrow. And so I'm not going to say a lot about it now, except to say that this is a physically meaningful model that describes the flow of fluid through a porous elastic network. We have this additional parameter, the permeability, that tells us something about the flow and tells us something about the porosity. And we can look at the creep data just as we do with the viscoelastic data, and it gives us a modulus, a Poisson's ratio, and this permeability. And there's way too much detail. I could give an entire talk about just how we do this and how we developed this in the early 2000s, but I'll skip over that for now because it is all available in the literature, and I can talk about it with you at the break. So here's we're comparing the microindentation, the nanoindentation. You can see the scales there are totally different in terms of the indentation depths, but we have these creep curves, and we can fit the creep curves. The fits are very good, and from that we get these 3 key material properties. So when we look at the data, and the reason there's so much data plotted on here is we were investigating the effect of one of the assumptions of the poroelastic model, which is how the Poisson's ratio is prior to the fluid flowing. So that's why there's 3 points for each thing. The bottom line is pretty clear though. When we do microindentation in blue or nanoindentation in red on the same sample, so we have 4 bone samples here, A, B, C, D. A and B were only tested at the microscale. C and D were tested at both the micro and the nanoscale. The modulus is about the same. And for these hydrated materials, again, the modulus is about 500 or so megapascals in the fully hydrated state, which equates to between 1.5 and 2 gigapascals. So we're still in the spherical indentation. These are different bones. This is a different study, completely independent of the ones I presented earlier. But for these hydrated bones, tested wet, viscoelastic or poroelastic, we're always getting about 2 gigapascals. So we're an order of magnitude below the typical values people see for dry Berkovich data. Okay, that's fine. All the data are about the same. Microindentation, nanoindentation show the same. What you get is Poisson's ratio, and it's way too complicated to go into at the moment about why we can measure Poisson's ratio with this approach, whereas you can't with traditional nanoindentation. I'm going to skip that because I want to really focus on the permeability. And so here's the results of the permeability. Yes?
One quick question. In the previous plot,
Yep.
it looks like there's not much anisotropy because it's not showing up any microstructural effect.
These are all tested in the same plane.
Same
So these are the, what is it, MC? They're like the ones Andy does, the MC3T, no.
Oh, third metacarpal.
The third metacarpal, thank you, yes. I'm not a bone expert, so the details of the bones are not my forte. They're all tested in the same orientation, so you wouldn't see any anisotropic effects. We get them, they're cross sections, we get them as discs and polish them up. But yes, that's a good point. As George showed, you can do beautiful work with anisotropy. The key thing here is that when we plot this permeability, which talks about the resistance of the fluid flow through the bone, we see a 2 to 3 order of magnitude difference between the results that we have from the microindentation and those that we have from the nanoindentation. And the permeabilities are much smaller for the nanoindentation, which means it's much more difficult for the fluid to flow. So this is interesting. As we go to larger scales, it's easier for the fluid to flow. We go to smaller scales, and it's much, much harder for the fluid to flow. It's not like this is an ambiguous difference at 2.5 orders of magnitude. And this is fundamentally interesting and tells us something about our microstructure. And in fact, what we can do is we can take a picture of this microstructure. So this is one of the bone samples we tested. The black circle in the middle is the size of the contact for what we call our nanoindentation experiments. And the size of the dashed lines are what we had for our microindentation experiments. And what you can see clearly on the bone bone sample there is that there are a lot of pores that are about the same size as our contact size for the nanoindentation. So when we're nanoindenting on basically solid material because we're choosing those spots because we have this view in the microscope of the nanoindenter, whereas when we do the microindentation, we have these larger holes. And what these different sizes of porosity are related to is how easy for the fluid to flow. So now our data make complete sense, because as we go to the very large contacts, we have lots of these macroscopic pores through which the fluid can flow, whereas when we go to our very small contacts and we're selecting where we're doing this so that we know we're not putting the indenter into one of these pores, then we get this much more difficult fluid flow. It's much more resistant because we're now not going through these big tunnels. We're going through the actual material itself. And the reason we don't see huge differences in the modulus between the two types of tests is, of course, because the total porosity is relatively small. We recently did some more visualization of this 2-scale porosity just to kind of get a better picture of it compared to my relatively crude cross-section, but you can see the same thing. So the smaller porosity shown there in the yellow called the lacunar canalicular porosity, and that's really the material porosity, whereas the larger scale porosity, that's the vascular porosity, that's where the blood flow can happen. And so we are picking up these two different porosities in bone, and just to finish up, we've actually found that even without having this huge difference between the nanoindentation and our much larger scale microindentation, just by varying the size of the indenter, we can pick this up on our nanoindentation specimens just by using different size spheres. So we have about 8 spherical indenters for our nanoindenter so that we can do these different experiments, and you can see some of the different contact sizes laid up on the cross-section there compared with the scales of the porosity. And the two curves at the bottom, I'm sorry it's a little bit small, but the two curves at the bottom are the creep curves for 2 tests on the same bone using the 2 different size indenters, where the larger size indenter is getting this larger scale porosity and the 10 to the minus 21 permeability. And when we go to the smaller scale, we're picking up only the material porosity, much less fluid flow, much less creep, and the same sort of difference in the permeability values for the different size scales. So my summary and conclusion is really this cautionary tale that bone properties, and I can imagine this is going to be true of many other biological materials that are not elastic plastic in nature, as you measure these things by nanoindentation, everything varies with the hydration state, with the indenter tip size and shape, with the data analysis method. And as I said, I can't emphasize enough, this is all based on the same elastic contact mechanics. And so this is really a question of how are we testing these materials appropriate to what their actual constitutive behavior is. If we're talking about materials that are fundamentally time-dependent, poroelastic, fluid flow, viscoelastic, and we try and analyze them with the exceptionally good methods for analyzing metals and ceramics, then we just don't know what we're getting. And so that is my conclusion. With that, I'm happy to take any questions.